Today is my pleasure to interview Dr. Charles Brenner, an expert in aging. And so, Dr. Brenner, thank you for being part of this episode. So, in hindsight of such a illustrious career, can we start with you taking us back and telling us about your background?
Sure. I'm a biochemist. Um, I went to undergraduate at Wesleyan University. I'm originally from New England. Graduated in 1983. My initial research experience was through flies. Then I spent about 5 years in industry before grad school. So I was in the San Francisco Bay Area at Chiron Corporation and at DNX Research Institute. Then 1988, started grad school at Stanford, Department of Biochemistry, got my PhD in cancer biology in 1993. My major thing that I did in graduate school was I purified and characterized an enzyme called KEX2 protease. So if you and your listeners have heard of PCSK9, which is a very important drug target in cholesterol regulation, or PC1/3, PACE, furin, actually, you hear about in virology. PC2, you hear about in insulin processing. These are enzymes that exist from yeast all the way to humans that have a role in hormone maturation. So I did a lot of work in that area. And then 1993, I moved back to the East Coast. I did a postdoc at Brandeis. I started working on nucleotide binding proteins. And 1996, started the Brenner Laboratory, my independent research career. I've been at 4 places now independently. I was initially at Thomas Jefferson University in Philly from 96 to
Mhm.
2003. And then 2003 to 2009, I was at Dartmouth. A lot of my well-known work occurred at Dartmouth. That's where I discovered the vitamin activity of nicotinamide riboside and the existence of a nicotinamide riboside NR pathway in organisms from yeast to humans. 2009, I was recruited to University of Iowa to be the head of the biochemistry department, and I was there for 11 years in Iowa City. My lab grew its program in NAD biology. We did some other things as well relating to DNA methylation and some other cool stuff. And then since 2020, I've been at City of Hope, which is in Los Angeles County, and they created a new department. You may know that City of Hope is well known in sort of two areas:
Mhm.
cancer, right? We have a freestanding comprehensive cancer center that's very well regarded, and then you may also know it's the home of the Arthur Riggs Diabetes and Metabolism Research Institute. So I'm the chair of a department in the Diabetes and Metabolism Research Institute, but the name of our department is the Department of Diabetes and Cancer Metabolism. So we're kind of like a link between the cancer focus, cancer research, and diabetes and metabolism research. And, you know, and the other thing that probably people know about me is that we're all NAD all the time, pretty much. We think about the central catalyst of metabolism, NAD, and we found ways in which it's dysregulated in cancer. We found it dysregulated in obesity, diabetes, and metabolic diseases, and so that's kind of our focus.
Yeah. So my next question is, you talked about you worked in different companies before you went to graduate school. So my question is, why did you think, go pursue like a career in the biotech field instead of, continuing your just right after bachelor, you do your PhD. So
Yeah. So, the typical track then was if you're a really good undergraduate, you go, into a PhD program directly. It's a little bit less common now. Now people tend to spend a certain amount of time doing something else, maybe gaining research experience. In my case, it's pretty simple. There's no one in my family that had a PhD, or really a doctorate of any kind. And, I graduated from college in 1983, and, I had, and I think parents will appreciate this. I had the notion that you go to college to not be a burden on your parents. And, if you've done a biology degree and it's early 1980s, and there's the birth of the biotechnology industry, then called the genetic engineering industry, by the way, then you get a job in that industry, right? So what I found, I was able to learn quite a bit in those 5 years, but I also learned that there was a ceiling, for me at not having a PhD. And so if I wanted to design experiments, if I wanted to run a research group, I needed to, to get a PhD, and in fact, do a postdoc as well. And so that was my route. But the other thing is that at all of my institutions, Jefferson, Dartmouth, University of Iowa, and City of Hope, I've had, discoveries that, were filed for patents and issued patents. And in fact, patents that have been licensed and have developed useful technology for people. And so I'm very interested in I'm very interested in how to use molecular biosciences to discover things, and to make life better for people.
Oh, so that's interesting. So, because I never knew there was like a ceiling in the biotech industry.
Oh, sure. I mean, all the bosses are PhDs and some MDs, and that's considered the basic requirement, of being a card carrying scientist, is you carry that card of, you have a PhD, and moreover, what did you do? What did you accomplish to get your PhD? Right. So I didn't just tell you, oh, I have a PhD from Stanford, right? I told you, as a graduate student at Stanford, I'm the one that purified Kex2 protease and characterized it. People know that, right? Mm-hmm. It's in my, the nice thing is that you do those things, no one can ever take it away from you, right? That, you're the person that did that work.
I see. Okay. So, let's talk about your research. You said you were a major researcher in the field of metabolism, specific NAD. So, for people who may not understand what it is, can you explain what it is?
Yeah. Okay. Definitely. So, here's the most relatable way to explain it, right? Is to start thinking about electricity. Okay. So, whether you want to think about an electric car or an electric toaster, electrical devices are run on high energy electrons, right? So toaster is plugged into the wall outlet. Electric car has a huge battery, but in both cases, there's copper wires going from the source of high energy electrons to all the parts that require them, right? And it's that current, it's that flow of high energy electrons that provides the power, right? Well, guess what? Living things run on high energy electrons also. And, the animal life, if you want to try to understand animal life, the thing that is a head scratcher initially for physicists thinking about living things is that the second law of thermodynamics, right, says that entropy is always going to be increasing, right? The tendency for disorder is so right. So you and I play pool today, right? And at the end of our playing pool, we rack the balls, right, on the table. And we take the triangle away, right? And then if we come back to that table, one day, 1 week, 1 year, 1 decade longer, right? With time and temperature and vibration, those balls are not going to be racked any longer, right? And they're going to become less ordered, right? And so physicists look at biology and they say, how is it that living things can kind of keep their order together? And of course, the answer is not by violating any physical laws, but by taking in energy, right? So an animal is not a closed entity. It takes in energy, right? And it runs metabolism. So we take in energy in the form of protein, fat, and carbohydrate, the things that have calories, are macronutrients, and we oxidize them. We burn them essentially. And ultimately, we're taking those macronutrients and we're converting them into carbon dioxide, water vapor, thermal energy, heat, right? But importantly, our enzymatic systems capture a great deal of the energy of oxidizing our fuels and are able to generate ATP with it and do biological work. Okay? And the molecules that capture the high energy electrons from our food, from our macronutrients, are called NAD coenzymes. So they are called NAD plus, NADH, NADP plus, and NADPH. And what my laboratory does is it works on how these 4 NAD coenzymes are formed inside of cells and how and all of the processes that regulate them, how they break down, how you take the energy from glucose oxidation in what's called the cytosol, and you get the high energy electrons into the mitochondria, where most of the ATP is made. That's a really interesting problem for us right now. So we're interested in the carriers of high energy electrons that make life possible.
So you talked about a metabolism. I think everyone's heard about food. That's where the word came from. Most of the people, they think, oh, I eat a burger, I get energy, that's metabolism. So is metabolism just about the food we intake, or is there something else in there?
Yeah, it's a great question. Metabolism is the ability to convert everything that we eat into everything that we are and do, right? So a full oxidation of our food all the way down to carbon dioxide and water vapor would only generate heat, and the cells would try to capture the energy in form of ATP in order to kind of like do work, right? Run your muscles. But the thing is, we start from a fertilized egg, right? And then we get to be a fully formed adult, right? Weighing 50 kilograms, 100 kilograms, whatever it is that we weigh, right? We've made all of our stuff. Our metabolism has made all of the protein, fat, carbohydrate, nucleic acids, plasma membranes, all of everything in our body we've made. Unless you have a Teflon hip or something like that, which an orthopedic surgeon installed in you, you've made everything in your body through metabolism. And so the partial breakdown of our food into components like pyruvate, lactate, alanine, acetyl-CoA, alpha ketoglutarate allows us to partially break our food down and then build things up, right? So we make our own testosterone, we make our own estrogen, we make our own nucleic acids. And those are the anabolic processes, where the processes we're building things up. And that's where we're using high energy electrons to make stuff and to repair stuff. Because again, in this process in which animals, especially when we're growing, when from the age of conception to, the peak of physical maturity, we're actually adding a lot of muscle, we're adding a lot of neurons, we're adding a lot of complexity, right? And that really is very energy dependent, energy demanding. It requires a lot of food intake. Like, think of the way that babies grow. They will double in size, right, over relatively short periods of time, infants. That requires a lot of energy, and that's capturing those high energy electrons into growth and anabolic processes.
So, you talked about how like our muscles are created from the energy that we intake from our food. As you get older, do you gain less muscle because less energy is coming in, or is that something?
Yeah, there's a program, there's a developmental program that kind of gets us to our peak size. By being a bodybuilder, you can fill out your frame more, or by being a calorie restrictor or living through famine or impoverishment, you can sort of not grow to your maximal size. Like right now, the people in the Korean Peninsula share a lot of genetic traits, but the people south of the demilitarized zone in South Korea are bigger than the people in North Korea. Why? Because their nutrition is better. So there's a genetic developmental program that kind of gets us to our adult size, right? And there's higher levels of androgens, there's higher levels of growth hormone in that process, right? Once we get to kind of our prime and our peak size, there's a kind of a degradation of some of those anabolic programs. And, frankly, it's harder to keep your stuff together, right? One of the ways of thinking about it is we have gene sets that, basically control what our potential is and what we're like. That's why identical twins, are so similar. They're not 100%, identical. Their lived experiences may be different, especially if they're, separated at birth or something like that. But and these things are environmentally influenced. But we have a gene set that gets to our potential, and evolution sort of doesn't care as much once we've reached our potential to reproduce and carry on our gene set. Now, humans are remarkably good agers, right? So if you look at animal longevity data, in general, the biggest animals live the longest periods of time. But that's not true in the primates, right? Because gorillas are bigger than humans are, but humans live longer than gorillas do. And it's thought that grandparenting in particular grandmothering, was so important in human development that grandparents have helped create an environment that ensures that their grandchildren are able to be successful, that they can reach reproductive maturity, and that they can have babies and carry on the gene set, right? And so humans live a very long time after our reproductive peak, and women, have an average onset of menopause at 51, right? And yet they are healthier than men in their 50s, 60s, 70s, and 80s, and tend to outlive men, even though they're beyond their reproductive capacity as individuals. The human gene set allows women pretty uniquely to be very healthy after they're reproductively competent. But generally in animals, animals don't generally live longer than they're capable of reproducing. So longevity is not a sort of directly selected trait. Does that make sense to you?
Yeah. Mm-hmm. Well, that's interesting because I remember I learned that someone told me that there were more women. Actually, math class, they said there are more women than males on this planet. So that may be why the aging, maybe why there are more
It,
females
you
than males.
have to look at the data, right? And you have to look at the percent female at live birth, right? And then you'd have to look at survival by decade, right? And then you'd have to think about whether it was, and then you'd have to try to figure out what the mechanism is. Like, is it that there's so many wars and there's a bias of more men, being in more dangerous situations, or is it an intrinsic health thing? But the data pretty clear for humans that women are healthier in their age.
So, Ahmed, you also discovered the NR kinase pathways, and why are these pathways so important to aging?
Yeah.
And can you talk more about what the so-called NR kinase is?
Yeah. So NR stands for nicotinamide riboside. I told you there are 4 NAD coenzymes, and the biggest piece of NAD that can get into a cell intact is something called nicotinamide riboside. And it was known that certain bacteria can use nicotinamide riboside to make NAD. That was known almost since the 1940s. But it was not thought to be an NAD precursor for what are called eukaryotic cells, for cells that have a nucleus, and that includes fungi, plants, and animals are all eukaryotes. And so in 2004, we showed that NR is what's called an NAD precursor. It can be used by yeast cells to make NAD. And we identified the nicotinamide riboside kinase, the NR kinase. And then we showed that there's NR kinases in mice and in humans. And I think the reason why the NR pathway is important is that I told you why NAD coenzymes are important, right? They literally carry the high energy electrons that make life possible, right? That make it possible for us to generate ATP from our fuels and to generate ourselves from our fuels and to repair ourselves. But the other thing that our laboratory has discovered is that there are many conditions of metabolic stress in which the NAD system is disturbed. So, for example, when you go out in the sunlight, ultraviolet radiation from the sun damages our DNA. And in order to repair that DNA, skin cells use NAD. And when you're exposed to sufficient noise to produce hearing loss, your NAD system in your cochlea, in your hearing organ, is disturbed. We've shown that when you overfeed mice and you develop obesity and type 2 diabetes in mice, their liver NAD system is disturbed. We showed that not only in mice, but in humans that have drinking problems with alcoholic liver disease, their NAD system is disturbed. And we know now that in heart failure, neurodegeneration, time zone disruption, coronavirus infection, inflammation, and postpartum, there's a disturbance in the NAD system. So when there's disturbances in the NAD system, you basically benefit greatly from taking NAD precursor vitamins. And two of the NAD precursor vitamins were discovered in 1938. NR is from our laboratory, Bieganowski and Brenner in 2004. And again, because NR is the biggest piece of NAD that can get into cells, cells like to ramp up the gene expression of the NR kinase genes when they need NAD. Not in every case, but for example, in heart failure, when NAD is under attack, the failing heart turns up the expression of nicotinamide riboside kinase. So it's telling us it's actually looking for NR in order to kind of replete, if that's a word you can grok, to rebuild and restore their NAD system. So NR is a special NAD precursor in that it costs less ATP for cells to use it, and kind of in an emergency situation, cells really depend upon the NR kinase system to restore their NAD.
So, you conducted the first human trial of the NR. So, what were some challenges about doing this human trial, and what did this trial demonstrate?
It's interesting, actually. So I was on the faculty at University of Iowa at the time, and we had developed what's called quantitative targeted metabolomics system. So, like if you want to measure all of the NAD metabolites quantitatively, like not just, oh, this one's up and this one's down, but if you want to have them on a what's called a molar scale, where you actually know the concentration of all of them, you need to develop a method for that, okay? And the method is something called LCMS. It stands for liquid chromatography mass spectrometry, tandem mass spectrometry, in fact. And then in our case, it's quantitative, so we have standards so that we know the amount of the metabolites. And so we had done that a bunch of times, right? We'd done it on liver samples, we'd done it on human tissue culture samples, and we were being supplied with clean nicotinamide riboside, right? And it was looking like there's going to be a demand for human beings to like take this substance, and that based upon what its activities were in like mouse models, preventing diseases and treating diseases, and in one case extending lifespan of wild type mice, there's going to be a demand for this compound, right? And so we needed to enroll some people, that we're going to give them NR. We're going to consent them, right? And say, would you agree to be in a study? You're going to take nicotinamide riboside. We're going to measure to make sure that there's no adverse events. And then we're going to measure, your blood NAD, right? And we're not telling them that it's going to cure some disease. In fact, they're healthy volunteers, right? And so, you have to get people on board for this, and you can't tell them that if it's successful, it's going to help them directly. You can only tell them you're going to help the arch of research, right? The arc of research, so that we're going to learn some things about safety, and then in the future, we're going to be able to use that to blah, 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 right? But first, you have to figure out, how do you extract, the human NAD metabolome from human blood? Because no one's done that before, right? So I said, okay, to my lab, you know what? You guys could take my blood. I'll do it. And I'll take the first, doses of nicotinamide riboside chloride, and you could take my blood. So there's something called the Institutional Review Board. And the Institutional Review Board, at a university is there to make sure that you don't do unethical research, right? That you have a properly executed consent form. You're not putting people in danger. You have people to monitor the adverse events and so forth and so on, right? So I write this protocol and I say, I, Charles Brenner, am going to consent myself, Charles Brenner, to take this vitamin, nicotinamide riboside, and folks are going to draw my blood, and then we're going to look at it. And it's going to be an N of 1 study. And I got back an interesting and surprising response from the chair of the IRB, who said, that's not human subjects research. That I'm not allowed to give it to anyone else, certainly not anyone in my lab or anybody in the Department of Biochemistry, of which I was the head of the department. But that it actually didn't require an IRB consent for me to give a compound presumed to be safe to myself. So I actually did an N of 1 experiment. It's described in the paper as healthy 52-year-old male, I think. And then from that one experiment, we established the time course. So it said, okay, there's going to be a peak rise in NAD at like 6 or 8 hours after ingestion. And you're going to get a good time course if you look at something like 1, 2.5 hours, 8 hours, 12 hours, 16 hours. You're going to see the whole arc of the NAD, blood NAD rising and falling. So that was kind of the first, episode. And then once we figured out how to extract it from using my blood samples, then we were able to enroll people and consent them and know at what time points we need to collect blood from them. So yeah, that was kind of a fun story. I'm not sure I've ever told that story before.
Actually, I think I had another scientist that did a test on himself, and he got yelled at by it was also Stanford, but he got yelled at by the I think the board again for not writing a form this time.
So you want to err on the side of disclosure, right? Mm-hmm. So if I had said that I was going to take an untested drug, I'm not sure they would have gone for it. Since I told them I was taking a newly discovered vitamin, they took my word for it, I guess, that we had sufficient animal safety and toxicology data that it wasn't going to, harm me. That and that is the ruling that they made. There are very famous cases in the scientific literature, like the discovery that that ulcer is not from worrying, right? Ulcer is from Helicobacter pylori, a bacterium. That was discovered by someone who ingested Helicobacter pylori, an induced ulcer in himself, and then treated it with an antibiotic. So that's a very famous story. That is something that I'm not sure you could do today in a U.S. University, because that is, if the person's hypothesis is correct, that was going to generate illness. And but in my case, I did it, and I'm pretty sure I still have the email, from the authority at University of Iowa.
Yeah, so then you should also show that NR increases the SIR2 activity. So if for anyone who's not familiar, who does not know
Are
about,
we going to get into SIR2 and sirtuins?
yeah. So what
Oh,
are
man.
sirtuins? And
Okay. So this, and let's try to do it quickly. I mean, but so SIR2. So SIR2 is a gene that was discovered by Jasper Rhine and Ira Herskowitz, and I think also Amar Klar in around 1975. And the SIR2 gene in yeast has a role in yeast mating type. Did you know that yeast has a mating type? Probably didn't. Yeah, like the normal wild type yeast, like Fleischmann's yeast, doesn't actually have a mating type. It's polyploid, but laboratory yeast strains come in two mating types that are called A and alpha. They're not male and female, but they're A and alpha. Mating type A makes a lipophilic mating pheromone. Mating type alpha makes a hydrophilic mating pheromone. They fuse together and they form a diploid zygote that can also sporulate. This is classical yeast molecular biology. And I did yeast molecular biology professionally from 1983 to 1988. So in my first 5 years, when I was in industry, I was doing yeast. And then, as I told you as well, the prohormone convertase that I purified and characterized was also in yeast when I was as a graduate student. So I know a lot about yeast. So SIR2 is a famous yeast gene. Okay, now in the late 1990s at MIT, in Dr. Leonard Guarenti's laboratory, a number of investigators, all essentially well known now. Kaberlein, Kennedy, Sinclair are the key people. They showed that extra copies of the same gene, SIR2, extend one model of yeast aging. Okay, that's true. It's kind of strain specific. There's yeast strains in which you don't get the effect of SIR2, so it's a little bit more complicated than that. And also, there's another way of measuring aging in yeast called chronological lifespan, in which extra copies of SIR2 are actually bad for aging. Like the yeast SIR2 deletion strains actually live longer chronologically. They will live for more days and weeks if they don't have SIR2 than if they do have SIR2. The Kennedy, Kaberlein, Sinclair, Guarenti type of aging is called replicative longevity. That's the number of times something called a mother cell will form a bud or a daughter cell. And that's more times if it has extra copies of SIR2. So the problem is that that's not a selected trait. Okay, that's not a selected trait. A selected trait is something that you can pass on to your kids, right? So if you're clever, right, and you have a high functioning brain, or if you have good visual acuity, and that advantages you, or you're really big and strong, or really good looking, if you have your nice body proportions, those are controlled by genes, and you can pass on those genes, right? But the thing about this yeast replicative longevity trait is that this trait only emerges after something like 20 generations of the oldest mother in the culture. So there's literally 1 out of 5 million cells in a flask, in a yeast culture, that has this advantage of extra copies of SIR2. You can regenerate the yeast culture with any one cell. Okay? So the idea that the yeast SIR2 gene was going to be an important gene in other forms of life by virtue of selection is pretty ridiculous and unreasonable on its face. Unfortunately, in the early 2000s, there are very highly cited non-replicable experiments that were done that suggested that animal SIR2 genes extend lifespan. So someone actually from the Guarenti lab, Tissenbaum and Guarenti, did experiments saying that extra copies of this worm SIR2 gene extend lifespan in worms. And there's experiments from the Mark Tatar lab with David Sinclair as a collaborator that seem to show that extra copies of the fly SIR2 gene extend lifespan in flies. Those experiments are not replicable. In fact, deletion of one copy of the fly SIR2 gene extends lifespan when the fly is restricted for amino acids, which is one of the ways that you can extend lifespan in flies. It's like calorie restriction kind of thing. So the problem is the proponents of this sirtuin hypothesis, this idea that sirtuin genes are conserved longevity genes, have propagated this idea for 25 years. Billions of dollars have been misallocated pursuing this idea. Totally bankrupt concepts, like the idea that resveratrol is a SIR2 or human SIRT1 activator that you should take in order to activate your sirtuin genes and is going to extend your lifespan. Those ideas stem from a line of research in which, yeah, SIR2 extended lifespan in one model of yeast aging, okay, but not the other model of yeast aging, and not reproducibly in any animal. And SIR2 is not, or SIRT1 and the other sirtuins are not replicably longevity genes in mice, and they're not longevity genes in humans. There's some data with a gene called SIRT6 that in some mouse backgrounds, you get some effect, but that is just an emergent property of SIRT6. And I contend that if you were to take any 7 genes, and by the way, there's SIRT1, 2, 3, 4, 5, 6, 7, and you exhaustively look for these kinds of phenotypes in 7 genes in multiple mouse strain backgrounds, you're going to find this type of effect. SIRT6 is a positive regulator of gluconeogenesis. Guess what? In mice Old mice have low blood glucose, so having more SIRT6 maintains their blood glucose and makes older mice healthier. But guess what, humans? As we get older, our blood glucose tends to rise,
Mm.
right? We have obesity and type 2 diabetes epidemic. And so the idea that SIRT6 activation is going to be good for humans is an absurdly unlikely idea. So sirtuins are not conserved longevity genes. Yes, my laboratory showed in 2007 that NR increases the activity of yeast SIR2 and extends yeast lifespan in that assay, but I never said that that's the use case for NR, is activating sirtuins. And I think that it's been a really huge contamination of the biomedical literature that has come from this sirtuin field.
So have you and Dr. Sinclair talked before about this?
Many times.
Okay.
I mean, I've known David since the mid 2000s. I was a consultant for his company, Certrus, who the purpose of which was to find activators of sirtuins. And this is the company that took a preparation of resveratrol, which they called SRT 501, and sold that technology to GSK, GlaxoSmithKline,
Mhm.
for $720 million. And then GSK, invested billions of dollars after that trying to, get, human clinical evidence in favor of the idea that SIRT activation would be a good thing, or that they might have actual new chemical entities that will activate it. And so I've known David for a very long time. And have we spoken one-to-one directly in the last couple of years? No. I've chaired a number of the biennial NAD meetings. I've invited him to NAD meetings. We've been friendly in the past. Do I think he is evidence-based? No. Do I think his book Lifespan is a helpful one? No, I've called it an important source of misinformation. So, but my critiques of it's not a personal thing. Science is supposed to be self-correcting. And so when you identify something that's wrong, as the sirtuin hypothesis is, or the idea that resveratrol is an activator, you're expected to call that out. And so, I very much expect him to retract incorrect claims in the literature and misleading papers. If he doesn't, that's bad on him. And I don't think in the future that he will be able to get trainees or collaborators if he goes around saying that he identified dominantly acting monogenic longevity genes in yeast that are longevity genes in humans, and that he still thinks that resveratrol is an activator. I think that's anathema to being evidence-based.
Mhm.
And I don't think it's a sustainable practice for him. Everybody makes mistakes, and so you have to, when you look at the body of evidence, you have to be able to say, okay, I was thinking it worked this way because of the evidence at the time, but now I've been shown these other data, and so now I'm willing to, revise my views and correct, publication. So I very much expect him to do that.
So are you saying Dr. Sinclair scammed 723 million from GlaxoSmithKline for some technology that doesn't really work?
He may have believed it at the time. And so I don't know if that's a scam in real time. At this point, it's a scam that he has a company that is still putting resveratrol into a daily supplement,
Really?
and he's still claiming that it activates SIRT1. He has a paper, on this that makes absolutely no sense, because his original paper in Nature, it's Howitt's et al. From I think 2003, where he identified resveratrol, and he said it was an activator of the yeast SIRT2 gene and the human SIRT1 enzyme, the enzyme in both cases, really. And within a year, two groups, in fact, one containing Matt Kaberlein and the other one containing Brian Kennedy, both of whom know David, and they worked with him in the Guarenti lab. Both of them showed that resveratrol doesn't activate SIR2. It essentially activates what's called a biochemical artifact. So it interacts with a fluorescent probe that was used to assay the SIR2 enzyme. And then, that should have killed the whole resveratrol idea. But David didn't give it up. And David came back, and he, in a large collaboration directing large numbers of people, he claimed that, oh, really, resveratrol and these other compounds that were identified at Sirtris do really directly bind to the SIRT1 enzyme. And he claims to identified a glutamine, a particular amino acid, in human SIRT1 that he says is responsible for its interaction with these so-called activators. But guess what? That amino acid is not even in the yeast enzyme. So if his newer paper on the mechanism of resveratrol depends upon this glutamine, then he couldn't have even discovered resveratrol in yeast the way he says that he did. So, and by the way, resveratrol doesn't extend yeast lifespan, and it doesn't extend worm lifespan either. So there's no one that I know in who's a careful, molecular cell biologist that believes those data. And it's shocking, actually, that he continues to defend it, because it's just undermining not only the credibility of the work coming out of his lab, but it undermines the credibility of the entire field of molecular gerontology, that you have Harvard professor claims this, that, and the other thing, and including things that are totally discredited.
Also, regarding Dr. Sinclair, I mean, I think Jeff Bezos invests like a lot of money into one of his aging startups. So my kind of question for you is, do you think these billionaires who try to invest in aging, maybe specifically SIRT1, like, do you think they're also kind of making the problem even worse?
No, I don't necessarily think investors are making the problem worse. I don't know, what Jeff Bezos invested in. But I do know that there are a number of billionaires that are invested in Altos Laboratories and other, so-called anti-aging startups. And I think that they may have been, in fact, misled by some people into thinking that aging is a much more easily solvable problem than it is. I was just at a conference in Shanghai a couple of weeks ago, and someone got up and said, aging and the rate of aging is modifiable. Okay, well, I agree with that, but guess what? aging is easily modifiable in terms of accelerating aging, right? So if we take up smoking cigarettes and drinking and not prioritizing sleep, and we eat, we take in way more calories than we expend, we can accelerate our aging, right? We can also improve our health and improve our aging trajectory. But the fact that the rate of aging is modifiable in terms of making it worse does not necessarily imply that you can reverse aging. And while there are many people that are buying, consumer aging tests that are going around claiming that they've reversed their age, that's not accompanied by, functional metrics. And Brian Johnson is measuring things like his VO2 max. And VO2 max is actually really important. But, but it's not the case that everything that Brian Johnson is doing is, is evidence-based. So his fitness, regimen is impressive, if obsessive. But some of the things that he's taking are not evidence-based, and some of the things that he's measuring don't tell him what he thinks they mean.
I think he wasted $2 million a year just for trying to de-age himself. That's what I heard from numerous articles. Man, I don't know. I feel like the guy's a bit kind of obsessive about de-aging. So.
Oh, he acknowledges that he's obsessive about it.
Okay.
No question.
So, do you think in the future humans can have can't be on this planet forever? Like we actually won't die, or do you think that's like a myth?
No, I don't think that we're approaching longevity escape velocity. I debated Aubrey de Grey on this topic. I just saw yesterday on YouTube a discussion. It was supposedly a debate that had a number of people at like a New York City longevity / DSci conference that were talking about longevity. And I was just surprised at the lack of diverse viewpoints, in the conversation. Like no one seemed to, everyone seemed to be stating truisms, like, because we believe that, structure, dictates function, right? So we don't have a supernatural view of living things, right? So a number of the speakers said that the aging, and by which they meant post reproductive maturity, right? So after we reach our reproductive maturity, the age-related decline, right? It is a degenerative process. There's accumulation of damage. There's an increase in entropy. And, more than one speaker said, it's a problem that the molecules are in the wrong places. And so if you can get the molecules back in the right places, then you would have, rejuvenated the person. So that's just like going back to the pool table. If you can rack the balls again, then you would have racked the balls again. They would be as good as they were when we initially racked them. But the level of complexity, though, comparing, what is it, 10 billiard balls on one plane in a simple shape to, with trilateral symmetry, by the way, to a human, the number of molecules in a human, the number of different cell types in a human, the number of trajectories of all of those molecules in a human. In theory, if you put it all together again, you would rejuvenate that person. You'd also erase their memories back to the point at which you put all of the molecules back together like that. But, it is a Humpty Dumpty problem, right? That you can break an egg, but we don't know how to put an egg back together. Now, can I say definitively that we can't? No, I can't say definitively that we can't, but I would say that death and taxes are things that you can pretty well bank on when it comes to
Yeah, man,
mammalian
you're a
life.
co-founder of Pro Health Span. So what were some challenges in running a startup that sells supplements?
So okay, let's say what Pro Healthspan is. So I'm the chief scientific advisor of Chromadex. And so Chromadex is the company that called me when I was on the faculty at the University of Iowa. And they said, we're really interested in that technology that you developed at Dartmouth for nutritional and pharmaceutical uses of nicotinamide riboside. Because like I said, NR was a known entity, but no one ever thought about using it in humans, right? No one knew that there was an NR pathway. So we had patents that were issued, to Dartmouth College with me as the inventor, that enabled commercialization of NR, and that commercialization was done by Chromadex, and Chromadex was at the time an ingredient company. So they sold NR to other companies. Some of those companies actually kind of coveted the intellectual property and then went to war against the Dartmouth patents when they couldn't, control that IP or get the ingredient on the terms that they wanted it. So that's kind of a sad story. But Rob Fried, who was an investor in Chromadex, wanted to create a finished goods company and make a package, a supplement that had Niagen, which was the ingredient, patented nicotinamide riboside chloride, and sell that as something called True Niagen with a catchphrase on it that Rob and I discussed, and we decided to trademark the term age better. Like, you're not going to reverse your age, right? You're not going to stop aging, but potentially with NR, you're going to support your resiliency, you're going to support your metabolism, and you're going to try to age better. And so Pro Healthspan was the entity that introduced True Niagen, the branded, Niagen finished product, and then Pro Healthspan was essentially acquired by Chromadex, and so it's now the Chromadex, brand of finished material. So I learned from that. I'm a full-time employee of City of Hope. I've always been a full-time employee of my, academic institution, whether it was Jefferson, Dartmouth, University of Iowa, or City of Hope. But this is a consulting activity where, I advise, companies that have technology that I'm interested in, of like how to communicate about it and what would be a good research direction to go.
So do these supplements go through the approval faster than drugs and vaccines, or is it the same?
Yes. So FDA, the F in FDA stands for food. And the food division of FDA covers food and supplements. Like supplements, if you go to the aisle in Whole Foods or grocery store, you see over the counter products. So that comes under the supervision of the FDA, but it's not the level of supervision for the drugs, right? So there's sort of like two designations that you get, and Chromadex did things the right way. So they did toxicology in in animals, and then put together a safety dossier and got a new dietary ingredient notification for Niagen, and then also got a generally regarded as safe notification. And so those are the two notifications that were received by Chromadex for our preparations of NR. Like, somebody else, some sort of pirate company that is making NR, we don't know what their manufacturing process is. There was a time when somebody else was making it, and there was toluene. There was an organic solvent that remained in their material. So the fact that NR has a new dietary ingredient notification and generally regarded as safe, that's for our particular company's preparation of that stuff that, is an indication of safety. And by the way, I will add that there is no NMN is a very closely related compound to NR, so it's NR with a phosphate on it. Sort of a weird thing to make as a supplement, because NMN has to kind of lose the phosphate. The phosphate has to come off before it goes into cells. As I said earlier, NR is the biggest piece of NAD that can get into cells. But since NMN was tested as a drug before anybody got a new dietary ingredient notification for NMN, NMN is not actually legally sold as a supplement in the United States. And anybody that is selling it, NMN in the United States, is not, like, whatever the level of safety review is that is required, that I described to you, the toxicology and the safety and the dossier for the new dietary ingredient that generally regarded as safe. Anybody that is selling NMN now basically doesn't care about that. And so what's in the bottle is going to surprise people. There's going to be toxins. There's going to be much less NMN than people think they're getting.
Yeah, so my next question is about, I think there's another, you probably know, Dr. Leonard Garante. He has his own dietary supplement company called Elysium Health. I've seen ads on Twitter. So what makes Chromadex different from Elysium Health?
Chromadex, licensed the technology, right? And by the way, Elysium, when they introduced their initial supplement basis, they had my patents on the bottle. You can Google that. The original patents on Elysium basis were my patents that were Charles Brenner is the inventor and Dartmouth College was the owner for nicotinamide riboside. And then there's a second ingredient in Elysium basis called pterostilbene, which is related to resveratrol, right? Which is this, non-evidence-based story about resveratrol activation. Pterostilbene is a blueberry-derived polyphenol that's related to resveratrol. It's more stable than resveratrol. And the idea was that pterostilbene is going to be like a better form of resveratrol. And Garante, by combining NR with resveratrol, he claims, that he's got two different ways to activate your sirtuins. Well, guess what? I just told you sirtuins are not longevity genes. His, classic paper on extension of lifespan by the worm SIR2 gene is not replicable, right? SIR2 genes are not longevity genes in mice. They're not longevity genes in worms. Biotech and pharma have spent billions of dollars on this failed sirtuin hypothesis. And then he comes out and says, we're going to activate your SIR2 genes with NR plus pterostilbene. Initially saying that he's doing it, it's sort of speaking out of two sides of his mouth, because he's saying that, this came out of my lab. He's saying this is like an MIT invention, putting together NR with pterostilbene. But if you look at the actual inventions, the actual patents, it was my IP. And it was some IP that came out of either Mississippi State or University of Mississippi, where the pterostilbene, IP came from. By the way, pterostilbene raises low density lipoprotein cholesterol, bad cholesterol in human beings. So there's no reason for a human being to take pterostilbene. So, I was offered very early on, before I knew that pterostilbene was going to be really bad for people. The Elysium actually offered me a consulting agreement. Chromadex didn't object to that. I didn't like the offer that they made to me, so I didn't get involved with them. But later on, they basically tried to, steal the IP, and they ordered material that they didn't pay for. You can actually Google this. I'll send you some links where you can learn more about the court cases and the battles between Elysium and Chromadex. But no, I don't have real fuzzy feelings towards Elysium Health.
Let's see. So why you decide to develop the pre-med curriculum?
So I was, as I said earlier, I was the head of a biochemistry department in the early 2010s. I was there 2009 to 2020, and University of Iowa, the biochem department kind of teaches biochemistry to all comers, like undergraduate non-majors who are mostly pre-meds, undergraduate majors, graduate students, medical students, and also dental, and at the time, pharmacy professional students. And then in addition to junior faculty and residents and fellows that want to learn some, biochemistry, we teach them as well. And so we're huge stakeholders in biochemistry education. And what we saw coming down the pike was a revision of the MCAT, the medical college test. And it was clear that there were going to be biochemistry questions on the MCAT. And yet they were not, the AAMC, the Association of American Medical Colleges, was not stipulating what the undergraduate curriculum should be. So I basically wrote some opinion pieces on how to revise the undergraduate curriculum.
Actually, so my pre-med friends wants me to ask you this. Did you make the curriculum recommendations more strict or more flexible?
Strict or more flexible? I don't know. So who makes the curriculum, is kind of the pre-med advising office at any, university comes up with recommendations of what you should be doing if you're pre-med. Right? My feeling about it was that you need to have molecular literacy, right? Not only as a doctor, as a future doctor, but as a citizen, as an educated person, right? And so here are some of the fun facts about medical and pre-medical education in the United States. See if I can remember the data. At the time that I wrote these essays, there were about 3 million new college freshmen per year in 2 and 4 year colleges in the United States, of whom something like 500,000 take general chemistry. So that's an indication that as many as 1 out of 6 college fresh people, first year students, may be interested in something like a pre-medical track. Okay. Out of those 500,000, about 165,000 take organic chemistry. So 2/3 of them have already decided to do something else, right? And then out of the 165,000, that take organic chemistry, there end up being something like, I can't remember, something like 50,000 medical school applicants for something like 20,000 seats in first year medical school classes in the US and Canada. And then of those 20,000, about 19,000 of them become doctors. So once you get into medical school, almost everyone will become an MD in 4 to 5 years, generally in 4 years, right? But of the 500,000 people that initially take general chem, very few of them are going to become doctors, right? They're going to find their passion, right? Whatever it is. So it's my feeling that first of all, we should do the biochemistry really early. I suggest we should do basically chemistry of functionalized carbon really early. Like freshman chemistry should be a type of organic, really a type of bioorganic that emphasizes carbonyl. So a lot of, functionalized carbon, less alkanes, alkenes, and alkynes, but you can teach kind of molecular hybridization, electron hybridization by thinking about carbonyl chemistry. And once you teach carbonyl chemistry, then you're ready to teach things that relate to the chemistry of life. Proteins and carbohydrates and fuel oxidation and lipids. But I'm not for piling on more and more and more semesters that are required. I actually think that people should take literature. People should take dance if they want to. People should take music and art and jazz and politics and history. So I don't actually want to load up undergraduates with semester upon semester of required courses. I try to guide, chemistry departments to think about the chemistry curriculum in a way that would serve molecular literacy, as well as better prepare people for careers in medicine, as well as biomedical research. But chemistry departments have kind of their own incentives to teach a lot of students, because that's the way they maintain a certain faculty size and TA size and size of the graduate program. So it's complicated.
All right. So is there any type of advice you would give to students or young people?
Find things that you love, read things critically, do research, talk to people whose points of view you don't understand yet. Those are pieces of advice that I think are worth listening to.
All right. Thank you, Dr. Brenner, for being part of this episode and giving me to talk about aging.